transduction pathway inside of the liver cell. And so we start the cell membrane right here, outside of the cell, inside the cell. The ligand, which is in this case epinephrine, can't go through the cell membrane, so it docks at an integral protein on the outside, or on the cell membrane. And a specific G protein then activates its peripheral, and in this case the GDP then gets the message and gets displaced by a GTP, which kind of activates transduction to occur. And right here from the epinephrine to the integral protein was reception of the message. And so the peripheral moves over to another protein on the cell membrane known as the adenylyl cyclase.
So the activated peripheral G protein causes the adenylyl cyclase to turn its ATP into CAMP, like this, and then the CAMP goes to the protein kinase, which has a regulatory and catalytic part, and then once they are attached to the regulatory part, the catalytic part is activated, and it brings the extra phosphate to the enzyme, in this case, which is glycogen phosphatase, activating the glycogen phosphatase, which then works on glycogen, breaks it up into pieces of glucose, and that is the response part of the signal transduction pathways.
Could be wrong about these, bro. Maybe I do.
There is a specific cellular response that occurs from a certain G protein, and the response is that it releases ions outside of the cell, regulating water concentrations inside and outside. And when cholera bacteria binds at the integral protein, it locks the G protein into its GTP form, thereby continuing the process and producing and exporting ions inside or outside the cell. The bad thing about this is that it never stops, and the process continues even after homeostasis has been reached. And with all the ions pushing outside the cell, water follows, and leading to dehydration of the cell.